So 11 point, does it say 11.3? It's 11.4. Sorry. Okay, 11.4 is sexual reproduction. So the first thing in this unit is gametogenesis, which is the production of gametes. So in humans, there's spermatogenesis and oogenesis. I don't know how to pronounce this. I'll say oogenesis. Spermatogenesis is the production of male gametes in the testes, and oogenesis is the production of female gametes in the ovaries. Both processes have the same basic stages. The first one is mitosis, then cell growth, then meiosis, and then differentiation. These are the same basic stages. Now, of course, they differ by a little bit depending on whether it's in the testes or the ovaries, because they'll have different processes. But yeah, so mitosis will generate a large number of diploid cells. Cell growth means that they'll have enough resources to undergo both divisions of meiosis. Meiosis will produce the haploid cells, and then differentiation will allow the haploid cells to develop into gametes with all the structures that are needed for fertilization. So the first thing we go through is stages of spermatogenesis. Like we talked about, spermatogenesis is the development of the sperm, which are these. So there's five main stages, and the sixth is how they detach, but 6 stages of spermatogenesis. The first is this is a part of a testes. So the outer layer, which is called the germinal epithelium cells. So these cells, this is the basement membrane. These cells are the germinal epithelium cells, which are all diploid cells. They will divide endlessly by mitosis to produce more diploid cells. So it'll keep growing into like these, right? And then the diploid cells will grow into larger and larger cells. So it starts with smaller cells and it'll grow into larger cells with more cytoplasm and will become primary spermatocytes. So the germinal epithelium cells by mitosis turn into primary spermatocytes. Then those primary spermatocytes will go through the first division of meiosis to produce 2 secondary spermatocytes. And then each of the secondary spermatocytes will carry out the second division to form 2 spermatids, which is the haploid cell. Are we all following? So just to go over it again, the germinal epithelium cells, which are the diploid cell, divide by mitosis to form more diploid cells, which then grow into primary spermatocytes, which is still a diploid cell. Then the primary spermatocyte goes through the first meiosis, where it becomes a haploid secondary spermatocyte, and then the secondary spermatocyte goes through the second division of meiosis to become 2 spermatids, which is still a haploid cell. Then spermatids become associated with nurse cells, which we call Sertoli cells. This is a name you need to remember. So the Sertoli cells help the spermatids, these cells, to develop into spermatozoa, which are still haploid cells. So this is an example of cell differentiation. So if you look at the shape, it goes from small cells to bigger cells, and then go back to average size cells. I would say they're bigger than the germinal epithelium cells, but they're slightly smaller than the diploid cell. And then as they become spermatids, then when spermatids become associated with nurse cells, they start to lengthen and then become into spermatozoa. That's the cell differentiation. So they lengthen and they change their structure to form spermatozoa. And then the spermatozoa, which are the sperm cells, they detach from the Sertoli cells and are eventually carried out of the testis by the fluid in the center of the seminiferous tubule. Did we all follow all the stages of spermatogenesis? Are there any questions regarding spermatogenesis? Because I know it's a difficult topic to undergo. Which is why does this all happen in the seminiferous tubule? I want to say no. This It happens in the, oh no, sorry, it does. I want to say yes. My bad. The 5 stages of spermatogenesis, the sixth is just the detachment, all occur in the wall of the seminiferous tubule. Yes. Okay. Are there any other questions? Or are we good? Good to go? Yeah, I'll assume the silence means good to go. Okay. I don't know if people still want to try this, but if you want to, label A to H, which is all the structures of a sperm. Of course, if you don't want to try it, it's okay. It's like, but it's good because they can ask this, they can ask like something like this in paper 1, where they can say what is structure B or what is structure C or whatever. So it's important to know the labelings, because in paper 1 oftentimes they can ask us label stuff, which makes it more difficult. Just gonna wait for everyone to answer. It's okay, we'll talk about it. If you don't know what it is, we'll talk about it. Okay, I think that's everyone who is answering, so I'll just go through all the things. A is acrosome, which contains enzymes that digest the zona pellucida around the egg. So the acrosome is what assists in the fertilization, as it digests the pathway for the egg to fertilize. B is the haploid nucleus that contains the 23 chromosomes that are passed from father to offspring. C is the tail that provides the basically enough movement to for the sperm to swim up the vagina, since it's it's anti-gravitational, so it needs enough energy. So the energy comes from fructose in the semen, but the tail provides the propulsion because it basically moves really quickly. D are protein fibers, which strengthen the tail. Sorry, it got cut off a little bit, but it's basically D are the protein fibers that strengthen the tail. E is microtubules in a 9+2 array. So they basically make the sperm tail beat from side to side and generate the force that propel the sperm. F is the helical mitochondria. So they produce the ATP by aerobic respiration, to supply energy for swimming and other processes in the sperm. These use the fructose that's in the semen around the sperm. G is the centriole, and H is the plasma membrane. I think a lot of us got everything. H is not the head though. It's because it's pointing at just the membrane, but it's not a bad guess. Acrosome, nucleus, tail. Yeah, everyone got everything except I think D and E, which is fair enough because D and E are very specific. But basically, the thin lines in the center are the microtubules, and the dark lines are the protein fibers that strengthen the tail. Okay, I'm gonna assume we're good with this. So the next is oogenesis. So oogenesis works in the same, it works in the same general way of gametogenesis, but it has slightly more steps. So firstly, in the ovaries, again, it's still germinal epithelium, which is a diploid cell, and they divide by mitosis to form more diploid cells. Then those grow into larger cells called primary oocytes. And primary oocytes will start the first division of meiosis, but in this case, they stop during prophase 1, where the primary oocyte and a single layer of follicle cells form a primary follicle. So instead of going from primary oocytes and going through the entire first division of meiosis, they stop at prophase 1 to form a primary follicle. And then when a baby girl is born, we contain about 400,000 primary follicles, which are these. But the process of, so they basically, follicles within the body stop at prophase 1, and the rest of the development is waited until the baby girl reaches the age of beginning the menstrual cycle. So every menstrual cycle, a few of the primary follicles will start to develop. So it'll be more than just one, multiple will start to develop. But so the primary oocyte will complete the entire first division of meiosis, forming 2 haploid nuclei. And the cytoplasm will be divided unequally. So there will be one larger secondary oocyte and a smaller polar cell. So when you compare it from oogenesis to spermatogenesis, we know that in spermatogenesis, it just divides into 2 secondary spermatocytes. In this, it forms, it's not divided equally. So that means that there's one large oocyte, but one small polar cell. And that's called the first polar cell. Then the secondary oocyte will start the second division of meiosis, but again, it will stop at prophase 2. So the follicle cells are proliferating and the follicular fluid forms at this point. So at this point, there's 3 layers of follicle cells. There's a secondary oocyte that stays at prophase 2, and there's follicular fluid within the follicle. So this is the developmental phase, and this will result in a mature follicle. And then when the mature follicle will burst, again, this is referring back to the menstrual cycle, when they burst, the egg that is released is actually still just a secondary oocyte. But then after fertilization, this secondary oocyte will complete its second division of meiosis to form an ovum. And then a second polar cell will be formed. So in oogenesis, basically, there will always be only one egg. There won't be multiple gametes formed. And that occurs because the cytoplasm will always divide unequally. So when it divides unequally the first time, it'll form the secondary oocyte and the polar cell. And then when it divides unequally the second time, when it goes to the second stage of meiosis after fertilization, it divides into an ovum and a second polar cell. So first and second polar bodies do not develop, and eventually they'll just disintegrate. But the egg will be an ovum. Are there questions regarding oogenesis? Oogenesis. Because I know this is even more complicated than spermatogenesis. After like the 5th step. Okay, so basically what happens is a follicle a follicle is formed when it stops at prophase 1. Yes? So when one reaches the menstrual age, and once the menstrual cycle starts, every menstrual cycle a few follicles will start to develop, and when the primary oocyte will go through will finish after prophase 1, will finish the first entire division of meiosis. That's when it divides into one polar cell and one secondary oocyte. So the secondary oocyte in this case is the small cell here, and the polar cell is even smaller over here. So that small black dot over there is the first polar cell, and the bigger circle is the secondary oocyte. That's how inaccurately they're divided. So when they're divided that unequally, only the secondary oocyte is actually capable of going through the process of meiosis again, and the first polar cell will disintegrate eventually, and then the secondary oocyte will begin the second division, and again, like earlier, it'll stop at the prophase and will not be dividing further until after fertilization. Before fertilization, it stops at prophase 2, and then that oocyte is what gets burst out of the follicle during ovulation, and that egg is still a secondary oocyte. And then that secondary oocyte, after fertilization, will complete its second division to form an ovum. So it'll have a haploid nucleus and a second polar cell. It'll again divide the same way it happens here. It'll be unequal, but there'll be one haploid cell, and then the other one will be a smaller polar cell or polar body, and then it'll eventually disintegrate as well. I know I just repeated the entire explanation, but I don't know if that made it any better. If you have anything specific that you don't understand, you can ask me and I can try to explain that bit. Okay. So when we say egg cell, we mean the secondary oocyte before it completes meiosis 2. Yes. The egg cell, when we talk about the menstrual cycle, when we talk about the egg cell being released from the follicle, the egg cell that's released is a secondary oocyte. And in simple words, yes. And what happens to the polar body produced after fertilization? It'll just disintegrate. Nothing of consequence will happen to it. It'll just disintegrate. Both of the polar bodies will disintegrate. So the follicles just keep on developing until it reaches the 3 parts of a mature follicle. So yeah, so certain follicles will keep developing, but only one follicle will actually be able to reach complete maturity every menstrual cycle. So every 28 days, only one follicle will actually reach the complete maturation. Yeah. Several may start to mature, but they won't mature completely. They'll mature in the next cycle. Are we good? Do some fail, or do they always just wait for the... Some might fail, but I don't think we need to know about them, really. You just need to know that oogenesis happens. But some might fail. Yeah, it's possible. Okay, cool. Do I assume that everyone's... I'm going to assume everyone's good. If you have any questions, feel free to ask. So, okay. So, another test thing. Try to figure out all the parts. This is an egg cell, mature egg cell. A mature human egg. Okay. Okay, waiting for, I think, 2 more people who are typing, and then I'll go for it. You have to double bar on either side, but it's okay. Just add another bar on either side. Yeah, there you go. Okay, so A is another haploid nucleus. It has only 23 chromosomes that are passed from mother to offspring. B is a cytoplasm, or the yolk, because if you think about it in terms of the like chicken eggs and stuff like that, it's the yolk. It contains the droplets of fat and other nutrients that are needed during the early stages of embryo development. C are the cortical granules. These are the things that harden the zona pellucida after fertilization has happened to prevent multiple fertilization and polyspermy. D is the zona pellucida. It protects the egg cell and restricts the entry of sperm. E is the layer of follicle cells, or the corona radiata. F is the first polar cell, so this is the first polar cell. It's not needed, it breaks down. G are two centrioles. Okay. Comparing spermatogenesis and oogenesis. So, firstly, millions of sperm are produced by men each day from puberty onwards, and they can be released frequently by ejaculation. But in females, from puberty until menopause, women who are not pregnant produce and release just one egg every 28 days. So it's not that many eggs. Nearly all the cytoplasm is removed during the latter stages of spermatogenesis, and sperm cells have barely any cytoplasm, but egg cells have the most amount of cytoplasm than any other human cell, because egg cells are the biggest cell in female human bodies. So egg cells have more cytoplasm than any other human cell, while sperm contain almost the least cytoplasm than any other human cell, besides, I believe, red blood cells. So the mitochondria of the zygote are derived from the cytoplasm of the egg cell and not the sperm cell. The egg cell destroys the helical mitochondria of the sperm after fertilization, so the mitochondria will be taken from the oocyte or the egg cell, the ovum, rather than the sperm. Okay, are we good in terms of this? Okay, good. Someone's typing for longer, so I'm going to presume there's a question, so I'll wait. The egg anatomy, yes, I can. There you go. Are we good? Okay, cool. So, fertilization. Now, fertilization is a 6-step process. First is the arrival of the sperm. So, sperm are attracted by a chemical signal and will swim up the oviduct. So, of course, they have to go through the vagina and then through the cervix and then through the uterus and then to the oviduct. But they swim up the oviduct to reach the egg. And for successful fertilization, there have to be many sperm that reach the egg. The second is binding. So the first sperm that breaks through the layers of follicle cells or through the sorry, I forgot the name. The corona radiata. The first sperm that breaks through the corona radiata will bind to the zona pellucida and trigger the acrosome reaction. So as soon as it binds, the acrosome reaction will be triggered. And the acrosome reaction basically is that the contents of the acrosome will be released and by the separation of the acrosomal cap from the sperm. So if we go back, we know that this is the acrosome and the acrosome cap. So the acrosome will digest the zona pellucida, but then the acrosome cap will separate and all the things that are contained within the acrosome will be let go into since it fuses to the zona pellucida, will be let go into the cytoplasm of the cell. Enzymes from the acrosome will digest a route for the sperm through the zona pellucida, which will allow the sperm nuclei to reach the plasma membrane of the egg. And then fusion means that the plasma membranes of the sperm and the egg will fuse, and the sperm nucleus will enter the egg and join the egg nucleus. Now, by joining, it doesn't mean that it's going to actually, the nucleus will fuse. The fusion occurs between the plasma membranes and the cells, but not between the actual nuclei. The next is cortical reaction. This is what prevents polyspermy. So small vesicles called cortical granules, after the sperm nucleus has made it into the cytoplasm, the cortical granules will move towards the plasma membrane and fuse with it, releasing their contents by exocytosis, and therefore causing cross-linking of glycoproteins in the zona pellucida, making it hard and preventing any other sperm to attach. And then 6 is when the nuclei for the sperm and egg, they don't fuse together, they stand together, and obviously the ovum finishes its meiosis, and then them together do carry out mitosis using the same centrioles and a spindle of microtubules, and then a 2-cell embryo will be produced. Are we good with the process of fertilization? They also destroy the sperm binding sites on the zona pellucida. Yes, they do. That's what the cortical granules do. Yes. Am I good to presume that we're good with this? Yes, it undergoes mitosis to form a 2-cell embryo and then a 4-cell embryo and then a blastocyst, which we will talk about. The zygote is the cell as a whole when the sperm and the egg nuclei are together, I believe. Yes. No. Yes. Acrosome are released and not the contents of the acrosome are released and not fused. Yes. The sperm will be fused with the plasma membrane of the sperm will fuse with the plasma membrane of the egg cell, but all the acrosome does is release its enzymes and therefore digest a direct a pathway through the zona pellucida. So here the acrosome will basically attack the zona pellucida, form a pathway here so that this plasma membrane can be fused with the plasma membrane from the sperm that's here, and then that will fuse together and then move the nuclei into here, closer to the main nucleus. Okay, so the next thing is early embryonic development. Now, the zygote that is produced by fertilization in the oviduct is a new human individual. And then it starts to divide by mitosis to form a 2-cell embryo, then a 4-cell embryo, and so on until a hollow ball of cells called a blastocyst is formed. So the zygote is basically when the nuclei from the sperm and eggs stand together within the egg cell. So basically what will happen is the sperm nuclei will be over here and they will stand together. This will finish its meiosis and they will stand together, and then that one entire cell will be considered the zygote. And then the zygote will undergo mitosis to form the 2-cell embryo and then the 4-cell embryo and then so on until this blastocyst is formed. So while the early stage of development in the embryo is going on, the embryo will be moving down the oviduct and into the uterus. And then when it's about 7 days old, the embryo will implant itself into the endometrium of the uterus, where it will continue to grow and develop. And then if implantation for some reason does not occur, then the embryo is not supplied with enough food and the pregnancy will not continue. So there's hormonal control of pregnancy and then the hormonal control of childbirth. So during pregnancy, human embryos or the blastocysts will secrete the hormone HCG, which is also known as the human chorionic gonadotrophin, from a very early stage. So what HCG does is stimulate the ovary to maintain the secretion of progesterone during the first 3 months of pregnancy, because there's nothing else to secrete the progesterone, because the corpus luteum has already disintegrated, so nothing will secrete progesterone. So the HCG will stimulate the ovary, which means progesterone will keep being produced, and that means that the uterus lining will continue to thicken so that it can support the embryo after implantation. And then by about the 12th week, which is considered the end of the first trimester, the ovary will stop secreting progesterone. But then by this time, a placenta will have developed, which means that the embryo will be well attached to the uterus, and therefore the placenta will take over the task of secreting the progesterone. This is also why we consider miscarriage to not be a risk after 12 weeks, which is after the first trimester, because the placenta has developed by that time. That means that the embryo has implanted successfully and effectively. And then, so the placenta will take over the task of secreting both progesterone and estrogen, which is needed to sustain the pregnancy until the time of childbirth. Blastocyst is a clump of embryos, yes, but it has a large space as well. The blastocyst, basically, because yeah, that's, I don't know how to explain it. It's a clump of the embryos, yeah. Hormonal control of childbirth. So during pregnancy, all throughout the 9 months, there will be rising levels of progesterone to ensure that the uterus will develop and sustain the fetus, and basically will thicken the uterine wall enough so that it and prevents contractions. Therefore, there won't be any spontaneous abortions or miscarriages. The level of progesterone will start to fall in the third trimester in the pregnancy, and more steeply shortly before the end of pregnancy. So when progesterone falls, it also allows the mother's body to secrete something called oxytocin and estrogen. So oxytocin is what triggers muscle contractions and uterine contractions, while estrogen is what causes an increase in the number of oxytocin receptors on the muscle in the uterus wall, which means that estrogen basically allows more oxytocin to be recepted, which means more contractions will occur as more oxytocin is produced. So that's why it's a positive feedback loop. This is considered a positive feedback loop. So when oxytocin binds to these receptors, it causes the muscle to contract, and then the contractions stimulate more oxytocin, and then the uterine contractions get stronger and stronger. So this is going to be, again, another positive feedback loop where more oxytocin means more contractions, and more contractions means more oxytocin, and therefore it becomes a loop. While the muscle of the wall of the uterus will contract, the cervix will relax and become wider and make the birth canal, and the amniotic sac will burst and the amniotic fluid will be released. And then finally, usually after many hours of contractions, the baby can be pushed out through the cervix and the vagina. Are there any questions before I go into placenta? And then that's it for all of this, and then we'll go into option C. Oxytocin also has to do with lactation. Yes, when you that's, I think, part of option D. It might be, I'm not sure, but when a mother is breastfeeding, oxytocin is produced even more. But that's, I mean, you don't need to know about that in terms of the scope of Unit 11. I will do option C after this. I mean, I'll take a break, but then I'll do it after the break. When is oxytocin released? Oxytocin is released at the end of, I know it's 11:15. I'm in the same time zone as you. It's okay. Yes, I will ping everyone when I start option C. You will know when option C starts, I promise. Oxytocin is released at the end of the gestation period, or the end of pregnancy, when labor starts to occur. The oxytocin is released basically at the end of pregnancy, when labor starts to begin. Is that okay, Steph? Okay, cool. Function of the placenta. So by the time the embryo is about 8 weeks old, it starts to develop bone tissue and is known from then onwards as a fetus. The fetus then develops its own placenta and umbilical cord. So the placenta is a disc-shaped structure with many projections called placental villi, which are embedded in the uterus wall. So the placenta is what allows the blood of the fetus to flow close to the blood of the mother and facilitate diffusion and the exchange of materials. So it's both for waste and for nutrition. So this is the structure of a placenta. There's the myometrium, which is the muscular wall of the uterus, which is used during childbirth. This is what contracts. There is the intervillous spaces, which is where the maternal blood will flow, brought by uterine arteries and carried away by uterine veins. Oxygenated, sorry, and then there's placental villi, which are small projections that have a large surface area for gas exchange and exchange of other nutritionist materials, but also of waste materials. And fetal blood will flow through the capillaries in the villi. Placenta, obviously we know that the placenta is a disc-shaped structure. This entire structure is the placenta. It is usually 185 millimeters in diameter and 20 millimeters thick when it's fully grown. And then the endometrium is the lining of the uterus. So myometrium is the wall, which is what contracts. If we talk about how placental the placental structure is, this is the umbilical cord, or the umbilical. There's umbilical vein, and there is one umbilical vein and two umbilical arteries. So the two umbilical arteries carry deoxygenated fetal blood from the fetus to the placenta, and then at the placenta, there diffusion occurs where oxygen is diffused into the into the fetal capillaries, or umbilical capillaries, while carbon dioxide is diffused back into the uterine veins. And so basically what happens is the uterine arteries will give the oxygen, and the uterine veins will take away the carbon dioxide, and the umbilical arteries will take the oxygenated blood back into the fetus. Does it produce hormones? Yes, progesterone, estrogen. Do we understand the structure of the placenta? Yes, I will be recording option C when I go through it. But I don't know if it will upload in time. But I will record it. I will do my best to record it. Yeah. Is there any questions regarding the main structure? And then I can go through how the transfer happens. Okay, I will presume silence means no. So, okay. Exchange of materials across the placenta. So, this is the placenta. This is the placental structure. There is mitochondria, and this is the chorion, which forms the placental barrier and controls what passes in each direction. There's, of course, microvilli, which will increase the surface area for the exchange. And there's mitochondria that provide ATP for active transport. There's also cytoplasm, which produces the estrogen and progesterone and secretes them into the maternal blood. And then there's a very small distance separating the maternal and fetal blood. So it's just the chorion that separates maternal and fetal blood. Now, this is a capillary. This carries fetal blood, and it's very close to the villus surface and has very thin walls of single cells. Now, this is inside a villus, I believe, and this is microvilli. So this is one villus of the cell. The chorion and the basement membrane is freely permeable, which basically makes it really easy for things to exchange within it. But the capillary will be really close to the chorion, and the mother's blood will be completely open in the intervillus space, which means that and this is a larger surface area, which means that there will be really fast diffusion between the two. And basically, so from the mother's blood, we get oxygen, glucose, lipids, water, minerals, vitamins, antibodies, hormones. Basically, anything that's useful to us goes into, diffuses through the chorion into the capillary. And then from the capillary, there's carbon dioxide, urea, hormones, and water, which are all the waste materials. And they will all make their way through the chorion into the maternal blood. And that will get carried away by uterine veins to be cleaned through the liver of the mother. Since babies don't have the same differentiated organs yet. That's pretty much it, I think. So if there are more specific questions, you can ask and I will answer. But I'm gonna take like a break for a little bit just so I can breathe and drink water. And then we will start and finish all of option C as fast as we can so that I can go to bed.